Alright guys, so this is meant to be an overview of this chapter. You'll see that we've kind of broken this down into the different sections that go along with the textbook. So what I did was I took this PowerPoint, which is out there if you just search Chapter 9 Campbell Biology. You'll find this PowerPoint, and then I modified this. Okay, overview, just to set the context for this. Life is all about doing work. So living things, they need energy from outside sources, and we call this free energy. We also call this available energy. Plants will make their own food, but then animals need to obtain their energy by eating plants or by eating other organisms. So big point here is just setting this entire chapter in the context of living things need available energy from some kind of source. And now this chapter is going to explain how they actually get energy from those sources. So there's the principle. Organisms need free energy to live, and here's how they get it in general. So energy flows into an ecosystem as sunlight. Photosynthesis then captures that light energy, and that process generates oxygen. It also makes organic molecules like glucose. Cells use the chemical energy stored in these organic molecules to regenerate ATP, which powers work. You'll also see this labeled as charging ATP or making ATP. What they're really saying is that the energy from the food that was in these organic food molecules is now being converted into a different chemical energy, and that powers up the cell's work. On a side note, the energy then leaves the system as heat. This is important. It's what's keeping your body alive. It also eventually will help us explain why there can't be as many organisms at the top of a trophic pyramid as at the bottom. But that's a couple chapters away. Here's a diagram showing the flow of energy in living things. So in any given ecosystem, we have light energy coming in. That energy is captured through photosynthesis, which builds up organic molecules like glucose. It also produces oxygen. These organic molecules and oxygen are used during cell respiration in the mitochondria, which creates this energy right here, ATP. By the way, when I say create, we've got to understand that nothing is really created or destroyed here. It's just transformed. So I'll try not to use it, but if I slip up, understand that nothing is really getting created. It's just being recharged or transformed. So we've made ATP, and then as the ATP gets used, it will end up leaving the system as heat, and then the byproducts of breaking down these organic molecules are used to do photosynthesis. So the matter travels in a cycle, energy flows in and out. Before we can get into the nitty-gritty, we got to go through some key background ideas. Otherwise, the stuff that follows just really won't make sense. Okay, the first concept here, and I've written these to correspond with the book sections, so you can go back and look at these, or rather they were already formatted that way. I just left them that way. So it's talking about catabolic pathways. Remember, catabolic means it's like a cat. It's something that is tearing something apart. So a catabolic pathway is reactions that are tearing molecules apart. These yield energy by oxidizing organic fuels. And we'll see this term again later, but essentially this chapter heading is saying that during any of these cell respiration processes that we talked about, it's really tearing large molecules apart and turning them into smaller ones that's giving us energy. So in other words, food molecules have high amounts of available free energy. Taking those apart and rearranging them allows organisms to utilize that free energy. And I'm just going to pull this highlighter up here just in case I need this in the future. Like I said, this is background info. So we need to understand this so we can understand the rest of the cell respiration process. And I've broken this section into 3 different parts so that we can kind of make some sense of it and categorize it. Part A is looking at the 3 different types of cell respiration. Part B is looking at these things called redox reactions and seeing their role. Part C is looking at how energy is harvested from the breakdown of food, so it's kind of a preview of the process. And then part D will really get into a preview of one specific process, which is aerobic cellular respiration. All right, part A, different types of cell respiration. When you looked at cell respiration before, maybe in your intro bio class, you might remember just seeing one equation or one type. Maybe you even talked about fermentation or heard the term anaerobic respiration. Well, we're going a little bit deeper in here, and we're taking that one idea of cell respiration and we're breaking it into 3 distinct types of reactions that could happen. So the big point in this first part here is that there are a lot of ways to break down food. Each way that you break down food is going to release energy. The amount of the energy differs. And there are 3 of these processes or types of cell respiration. I've got them ranked from most energy to least. The first is aerobic cellular respiration. People frequently just say cell respiration when they mean this one. The second one is anaerobic cellular respiration, which we will refer to as anaerobic cellular respiration. The third type is fermentation. And you will frequently see people list these 2 and use them as synonyms. So they'll say anaerobic cell respiration when they really mean fermentation. It's important for us that we don't get those confused, but that will make sense later on. If this part's confusing you, don't worry about it, we'll get to it. So for all 3 of these types, the bottom line is that they take the available energy in food and they use it to charge up cellular batteries. For example, ATP, that is the strongest cellular battery that we talk about. In this first type, aerobic respiration, like I said, typically called cell respiration, organic molecules are consumed and oxygen is also consumed. This one is by far the most energy efficient per glucose. And when I say energy efficient, understand that that's just in normal cell conditions. So that one makes the most energy per glucose. Anaerobic respiration is similar to this top one, but true anaerobic respiration, it uses compounds other than oxygen. That should have been over there, sorry. So it'll use things like sulfur and nitrate. In that process of anaerobic respiration, that's going to yield 2 to 32 ATPs per glucose. There's a big range there because it depends on what's used, where it happens, and which organism is doing it. The last type of cell respiration is fermentation. This one, it splits glucose into this other compound called pyruvate. It yields 2 ATPs per glucose. So by far the least energy productive in terms of ATP of the 3 types. All right, all 3 of these processes are catabolic because they're tearing apart large molecules. They're also exergonic because they are releasing free energy. Before we get into talking about this overall summary equation for cell respiration, there's a couple of things you should know. So like I said, technically when somebody says cellular respiration, that means both aerobic and anaerobic. However, usually they actually mean aerobic respiration. And like I said before, fermentation and anaerobic respiration, they are different, but a lot of sources, if you're looking at images or if you're looking at videos, will use those terms interchangeably. And typically, they're really referring to fermentation, not so much anaerobic respiration. Because this one, in the cases that we talk about, is fairly rare. Whereas fermentation tends to be a much more common thing that we talk about. Another point here, all foods, so we know about these macromolecules, carbs, fats, and proteins. They can all get consumed as fuel. However, you may have noticed this, the CR equation focuses on glucose because it's easier to follow. Here's the overall equation. You need to memorize this and you need to know it both ways, meaning you need to understand it in terms of the chemical formulas. You also need to know it in terms of the names. So on the reactant side, we have glucose plus 6 oxygen molecules. So 1 glucose molecule plus 6 oxygen molecules yields 6 carbon dioxide molecules plus 6 water molecules plus energy in the forms of ATP and heat. Commit it to memory. All right, part B of the background here, we are looking at these things called redox reactions and looking at their role throughout each of the types of cell respiration. Big ideas here, substances can be reduced or oxidized. You're going to see these terms over and over again in this unit and in the next one. And the reason why they're important is because if we can follow the electrons, we can really follow the energy. So redox reactions, they get their name because something is always reduced, something is always oxidized. So redox, reduced, oxidized. The significance, why bother knowing these? So if we follow this transfer of electrons, it's really that transfer that releases the energy that's stored in organic molecules. So that released energy is ultimately used to make ATP. Bottom line is if you know which compounds are being reduced and oxidized, that'll show you where the energy is going. Okay, the principle of redox in general, because you will need to know this, you might come across it in a few different spots. In general, a redox reaction is a reaction that transfers electrons between reactants. They're also called oxidation reduction reactions or redox reactions. This process of oxidation means one substance loses an electron. We say that substance has been oxidized. Reduction is the opposite. It means that a substance gains an electron. And it gets its name because the amount of positive charge is reduced. So we say that the substance was reduced. Okay, here's an example, sodium and chlorine. So sodium becomes oxidized, it lost an electron. Chlorine becomes reduced, it gains an electron. This is kind of a general formula. So this is substance X, substance Y. Substance X once had a lovely electron. It's been oxidized because substance Y has stolen its electron. So the electron, boom, went over here. X has been oxidized, Y has been reduced. All right, for knowing the terminology of redox, and this is really important just for the sake of not getting confused when you see it. The electron donor, in other words, the substance that was giving the electron, is called the reducing agent because it has reduced something else. The electron receptor is called the oxidizing agent. Another thing to point out here, because this can be confusing, technically some redox reactions don't show a transfer of electrons, or they don't actually transfer a number of electrons. However, they change the electron sharing in the covalent bonds. Here's an example. So here we have methane, and you can see in here that we have the electrons, and they are kind of being shared equally-ish between the carbon and the hydrogen. So this is pretty much a covalent bond here. And then we have these oxygen atoms, which you can see are kind of sharing the electrons right here in the middle. Now, according to the diagram, if we look at the carbon here, this carbon from the methane is about to become oxidized. So if you take a look at this carbon was once really close to these electrons. Well, now the carbon is really far away from these electrons. The electrons are being unfairly shared by oxygen in a polar covalent bond. So in this case, we would say that the carbon from methane has become oxidized. Boom. And on the other hand, the oxygen here now has a greater share. See, the electrons are closer to this oxygen than they were over here. So we now say that this oxygen has been reduced. All right, let's run through a couple of these in context, because really this is where it's more important to know what these terms mean. So organic compounds like glucose, they get oxidized during cell respiration as they get broken down. And things during cell respiration that get reduced are really the two most popular batteries that we talk about, NAD+ and FAD. So NAD+ gets reduced to NADH, FAD gets reduced to FADH2. Now, if we make this go backwards, so from FADH2 to FAD, would that be reduction or oxidation? That would be oxidation. Same thing right here. If we go from NADH backwards to NAD+, then that would be oxidation as well. Another spot where you're going to see reduction come up is at the end of aerobic cellular respiration, when oxygen gets reduced, meaning that this oxygen receives the electrons from the electron transport chain. Another helpful hint, you might have noticed this on the last slide. We may not actually see the electrons in the equations, so it might not always be written where something has a positive charge or a negative charge. So instead, what you're looking for, at least in the case of NAD and plus and FAD, is a transfer of hydrogen. So the electron is traveling with the hydrogen. So as the hydrogen moves over to something, it's actually been reduced. So here's NAD plus. The hydrogen has joined and it's brought along its silent electron that we can't see. So NAD plus has been reduced. FAD, same thing, it's coming over here. These hydrogens have joined it, and so now it's carrying with it its silent electron partners, the two H's are. So if we back up and look at the overall equation, you can look and see that in this case the carbon from glucose has been oxidized and the oxygen has become reduced. In these cases, oxygen is almost always going to be reduced. Like in all the cases we look at, you can assume that oxygen is the one being reduced. And then if you look at these and you're kind of getting lost and saying like, I don't know where the electrons are, just look, this carbon is now really close to two oxygen molecules. So it's buddied up with these oxygen molecules. So we could say this carbon has been oxidized. All right, part C of this background information. This is looking at how energy specifically is harvested from the breakdown of food. So food molecules, they get gradually broken apart or oxidized. This gradual step-by-step program is important because the alternative would be to simply blow it up, which creates a lot of released energy, but it's not really useful for life. So if we look at this, and this is kind of how the book will phrase it, a stepwise energy harvest via NAD plus in the electron transport chain. Here's really what they're talking about. In cell respiration, the glucose and other molecules, they get broken down in a series of steps. The electrons from those organic compounds are usually transferred to NAD+, which is a coenzyme. If that part throws you off, ignore it to start with. As an electron acceptor, NAD+ functions as an oxidizing agent, to use our new vocabulary. Each NAD+, that is essentially a stored energy, a stored battery that's tapped into to synthesize ATP. So this example really just getting at that idea that reduction and oxidation are important. When you see NAD+, that's getting reduced so that I can go on and reduce other things. And here's just another equation showing the same thing. So there's NAD+ gets reduced to NADH. Okay, a couple points here when we see NAD+ being reduced. This one, it's passing the electrons. Eventually, it's going to pass the electrons to this thing called the electron transport chain. You'll see that later on. Unlike an uncontrolled reaction, the electron transport chain passes those electrons in a series of steps instead of just one explosive boom. And we'd say that oxygen pulls the electrons down the chain, you could say in an energy-yielding tumble. That energy is used to regenerate ATP. Here we have two diagrams that are kind of comparing and contrasting the two different reactions, two very different reactions. So in this one, we've got hydrogen gas and oxygen. This is not in a living thing. This is just to show you what's going on. So if you had hydrogen gas and oxygen gas, you lit a match, boom, you'd get a big explosion. And we would release all the free energy that these two once had, now that they've been combined into a stable molecule. So we've released a lot of free energy. Compare that to cell respiration. where we've got two hydrogens in the form of NADH and we have oxygen down here. Well, instead of all the energy of an explosion coming out at once and blowing up our cells, they get released gradually, almost like you could picture water falling down these water turbines, gradually producing more and more ATP. Phosphorylation is another process that you're going to run into in this section, and it's talking about the transfer of energy between molecules. And what it's really talking about is moving this phosphate group. So here we have ADP. So there's the adenosine molecule, diphosphate, di for 2, so it has 2 of these phosphate groups. We phosphorylate it, add another phosphate to it, and it becomes ATP, adenosine triphosphate. That might be shown in a diagram like this. This is actually the opposite. So here's glucose. ATP drops off a phosphate onto that glucose and becomes ADP. So in this diagram here, we would say that ATP phosphorylated glucose and became ADP. Those terms, or the term phosphorylation, you're going to see in 2 different places. You're going to see oxidative phosphorylation. That is most of the ATP created by cell respiration. And that one is this big complex process that we'll talk about later. The other way that you'll see phosphorylation is during substrate level phosphorylation. And what's happening there is you have some kind of substrate, in this case, whatever this is with the green dots, and then that substrate goes up to an enzyme. The enzyme takes the phosphate group that was on the substrate and puts it right onto ADP. So ADP gets phosphorylated to ATP. That is substrate level phosphorylation. All right, last idea, or at least second to last, I can't remember, of the background information here is reviewing one specific process. This process of aerobic cell respiration. This is going to happen in 3 distinct stages and in multiple locations. Stage 1 is glycolysis. Stage 2 is the Krebs cycle or citric acid cycle, also called pyruvate oxidation. Stage 3 is oxidative phosphorylation, aka the electron transport chain plus chemiosmosis. So the book does a number of things to help try to keep these straight. They'll show glycolysis in that kind of teal color. They'll show Krebs in the citric acid cycle in the salmon color, and then oxidative phosphorylation in that violet color. All right, so right now in this figure, we're looking at the cell. So we'll imagine that this is our cell with a teeny tiny nucleus over here. So in the cytoplasm of the cell, the process of glycolysis takes place. Then the products of glycolysis move into the mitochondria, and in this space here in the middle of the mitochondrion, called the matrix of the mitochondrion, the citric acid cycle or Krebs cycle takes place. This diagram is really nifty. I would recommend pulling up the full thing and then studying it. Then the final stage here, oxidative phosphorylation, the electron transport chain, chemiosmosis, that happens in the inner membrane space, which is this space right here that I'm coloring with red. Okay, so we went through the background information, and now we're starting to get into the nitty-gritty. We said there are 3 processes or 3 types of cell respiration. We're going to focus on one of them right now, aerobic cellular respiration. So for following this process, it starts off with glycolysis. During glycolysis, we are harvesting chemical energy by oxidizing glucose to pyruvate. So glycolysis, glyco refers to the glucose, lysis refers to splitting, so it's literally the splitting of sugar. Glycolysis breaks glucose, one molecule, down into 2 of these pyruvates, so notice there's 2 of those. So the input is glucose, the output here is pyruvate. This stage is happening in the cytoplasm. So here's our lovely cell. Cytoplasm is all the goop that's inside the cell. So we're not in the mitochondria yet. All right, these diagrams you're going to find in your book, and it's not that important to memorize them. It is important to be able to describe them and understand how people would talk about them. So here we have glucose, and then the arrows are showing you, the black arrows are showing you that it goes in as glucose and it comes out as pyruvate. This diagram, they've broken it down into 3 phases. So they have the energy investment phase, they have the energy payoff phase, and then they have kind of the net or the overall effect. So as we go from glucose to pyruvates, you'll notice that ATP is used to phosphorylate the glucose. So you'd be expected to understand that ATP is phosphorylating glucose. I know that because it starts as ATP, adenosine triphosphate, and then it ends as ADP plus 2 phosphate groups. So those phosphates have come off and been attached to the glucose. During this energy payoff phase, we have ADP and 4 of these phosphates. And in this case, ADP has been phosphorylated to ATP. And then here we've got 2 of these NAD pluses. We are adding electrons, we are adding hydrogens, and we end up with NADH. So what process has happened from NAD plus to NADH? That's right, it's been reduced. So NAD plus has been reduced to NADH. So you'd be expected to be able to read through a diagram like this and then maybe answer some questions like, during which of these steps is ATP phosphorylating glucose? And you'd have to say, well, it's this one right here. Or during which of these steps do we see a redox reaction? In which case you would say this one right here. As far as keeping track of what's going on overall, this is a nifty little summary down here. So glucose came in, pyruvate came out. If we're looking just at the carbons, that's where the carbons have gone. So we had 6 carbons in glucose. We now have 2 of these pyruvate molecules. Each pyruvate has 3 carbons in it. So one glucose had 6 of these little buggers. Oh boy, I'm going to run out of room. Now we've made 2 of these pyruvates. I've got to stop drawing these because this is just not that great. We also had 4 ATP formed, 2 ATP were used up right there. So we have a net of 2 ATP made. We also have 2 NADs that were coming in, and then we've got 2 NADHs that are coming out. All right, so that was the first stage of aerobic cellular respiration, glycolysis. Now we're on to the second stage, which is looking at this thing called the Krebs cycle. So here we go. We have pyruvate. Notice we've got our 3 carbons right here. Boom. And this pyruvate, in the presence of oxygen, is going to get moved inside the mitochondria. So it once was, here's our cell, it once was outside for glycolysis, now it's moving inside into the matrix specifically. So in our diagram here, we have the cytosol, the cytoplasm, pyruvate moving in, and you'll notice that it is being converted. During this citric acid cycle, you'll notice that we have the complete breakdown of pyruvate. So for this to happen, we have to oxidize the organic fuels from pyruvate. This entire overall process is going to end up creating 1 ATP, 3 NADHs, and 1 FADH2s per turn of the cycle. It takes 2 turns of that cycle to make 1 glucose molecule get completely oxidized. And we're getting that because it takes 1 turn to oxidize a pyruvate, and there are 2 pyruvates that were formed during glycolysis. This process also regenerates the starting materials. So you'll see that term acetate. Now here we've got our pyruvates coming in, and in this diagram it would be expected that you could describe what's happening. So NAD plus is being reduced to NADH. The carbons here, one is leaving as a CO2. This other molecule here called CoA is coming in and joining in as acetyl-CoA. In this diagram, we have the original carbons shown in gray here. So you'll notice one of the original carbons has left. When I say original carbons, I mean the ones that were starting as glucose. And now we've got the two carbons that are left joined up with this other molecule called acetyl-CoA, and they go through this thing called the citric acid cycle. As they go through the cycle, those original carbons coming in as glucose will leave as CO2, and one turn of this cycle is going to create NADH, 3 of them, 1 ATP, and 1 FADH2. When we say create, of course, we're again not saying that something is actually just magically made. In these cases, specifically, NAD plus gets reduced, ADP gets phosphorylated, FAD gets reduced. In other terms, ATP gets charged up, NADH gets charged up, FADH gets charged up. We're charging up some batteries here. If we want to look at that in more detail and see exactly where those carbons are going, this one is a good diagram for following that. So here we started with these red carbons that had originally come in from glucose. Remember, one of the red carbons from the original glucose has already left as CO2. That process happened just to turn this into acetyl-CoA. Now these other two red carbons, you can see where they are going around in the cycle, and then eventually they hop off, which would be in between these two. Oops, I'm sorry. There we go. So I'm looking at them right here. So they're used. Those two red carbons are used to make a product, and then they're showing them leaving here in blue. All right. During oxidative phosphorylation, so this is the big payoff phase. Chemiosmosis gets coupled up with electron transport to end up making tons of ATP. So the available energy that originally came in from food, from glucose, is now stored in NADH and FADH2. These are both called electron carriers or batteries. During this last phase here, oxidative phosphorylation, these two batteries donate their electrons to this big thing called the electron transport chain, and that powers ATP synthesis. So the pathway of the electron transport chain. This chain is in the inner membrane of the mitochondrion. So citric acid cycle, we were in the matrix. Now we're talking about this space, this membrane, and this space right here. So if we zoom in, we're looking at that particular membrane, which would be right here. And then we're looking specifically at these proteins shown in greenish blue here. Those are the proteins of the electron transport chain. So most of the chain's components are proteins, and they exist in these things called multi-protein complexes because you got a bunch of proteins and they're all kind of stuck together. So these carriers, they're called, these proteins, they alternate between being reduced and being oxidized as they accept and donate electrons. So this is showing NADH comes and it reduces this protein right here. The arrow is showing the electron flowing to the next protein. So the next protein gets reduced, then the next one gets reduced, then the next one, then the next one. Eventually, those electrons are going to end up reducing oxygen to H2O. So that's our final electron acceptor, it's called. This pathway of electron transport, it helps to have kind of an overview here, and we'll just see how that's shown in a few different diagrams. So you might have seen this diagram right here. It looks a lot scarier than it actually is. This says free energy relative to oxygen. So what this has done is it's taken these proteins right here that were shown in green, and now it's kind of lined them up relative to the amount of free energy that the electron will have when it's with them. So it's shown to be like the electron is like a ball rolling downhill until it finally gets to the final electron acceptor, oxygen. All right, couple key details about the electron transport chain. The electrons get transferred from NADH or FADH2 to proteins in that chain. The electrons are going to pass between many proteins. That includes ones called cytochromes. And finally, they end up in oxygen. Remember, oxygen is really good at grabbing electrons, so it makes for a really good final electron acceptor because it is so electronegative. It really, really likes electrons. This process, it does not generate any ATP on its own. Instead, that movement of the electrons, it pays to pump hydrogens into the inner membrane space. So we've only talked about one part of this, the electrons moving down this chain. Those hydrogens, once they're in the inner membrane space, are used to charge up ATP, and that's what we'll go over next. So this first part was all electron transport chain. Now we'll go into the second part, which is chemiosmosis. So it helps to look at this diagram. Again, we're looking at this particular spot right here in the mitochondria. So that's called the inner, that's called the inner membrane. So here we have the matrix of the mitochondria, and then here we have the inner membrane space. Now that we've kind of gotten situated in terms of where this stuff is happening, what you're seeing here is that as these molecules, NADH and FADH2, drop off their electrons or reduce the proteins that are in this protein, the electron transport chain. So as they drop off their electrons, that pays to have hydrogens pumped from this side in the mitochondrial matrix into this place, the inner membrane space, which again is shown right here in black. And so these are dropping off their energy. By dropping off their energy, we mean they are reducing the proteins. And then what this is showing here in red is the flow of free energy, the flow of these electrons going down the electron transport chain, eventually ending up here in water, here with oxygen and forming water. While that happens, it pays to pump these hydrogen ions into this inner membrane space. So notice we've got a lot of these hydrogens now in the inner membrane space, a very high concentration of hydrogens. In the matrix, we have a very low concentration. Okay, now that we've built up a bunch of these hydrogens, a really high concentration of these hydrogens in the intermembrane space here in black, these hydrogens, they don't want to stay there. They want to flow back down from this high concentration where they're all packed in down to a low concentration where they're not as packed, back in the mitochondrial matrix. So they start to flow back across the membrane, passing through this specific protein right here called ATP synthase. This ATP synthase protein uses the exergonic flow of these hydrogens going down their gradient to drive the phosphorylation of ATP. So notice we have ADP plus a phosphate group, so ATP is being charged here. So this part is pretty cool. As the hydrogens come through here, they actually turn this thing, and that phosphorylates ADP into ATP. So this is kind of like a giant water wheel. The hydrogens come down through there, they turn it, and it phosphorylates and makes ATP. So we say this is an example of chemiosmosis. We looked at osmosis before, which is where you have a flow of water from one area to another. Well, this is chemiosmosis because it's the flow of a chemical. And this flow of this chemical, when I say chemical, I mean these protons, that is used in order to drive cellular work. And we would say that this is a proton gradient or a hydrogen gradient, because we have really high concentration over here in yellow on this side, and then really low concentration over here in the matrix. And that's being used to make ATP. You could also say that this entire thing coupled together is oxidative phosphorylation, because we are using oxygen right here in order to drive this process of phosphorylating ATP. So that's where this gets its name from. Again, just to back up and look at the steps. So overall, we call this entire picture oxidative phosphorylation because oxygen is being used to phosphorylate ATP. But if we broke that into steps, we have the electron transport chain. That's this step right here, where we have NADH and FADH2 reducing these proteins and creating this gradient, and then we have chemiosmosis. Okay, a couple more details about chemiosmosis. This proton gradient is referred to as a proton motive force, and you might see that again when we get into photosynthesis. You might also see that again when we talk about cell transport. All right, so we've been getting into the nitty-gritty. Let's back off now and kind of look at energy overall. If we just looked at energy. So energy during aerobic cell respiration, so during the steps that we were just following, typically follows this sequence. So it starts out in the bonds of glucose. The energy then flows into the bonds of ATP, NADH, and FADH2. These two use that energy and use it to charge or power up the electron transport chain. This then uses the energy to create this proton motive force, which allows the hydrogens to go through chemiosmosis, and then the energy ends up in ATP. And this is the big payoff, the jackpot. This is where we get to those 32 ATP. So even though it looks like we're making a lot of ATP, we've got to recognize that a lot of the energy that is in glucose is not actually getting transferred. It gets lost as heat. And then beyond that, you'll also notice, you'll see kind of a wide range of ATPs listed. If you look at different sources, you'll see 30 to 38 ATPs listed per glucose molecule. And the reason why, part of it is unknown, and part of it just comes down to how efficient the process was operating in those cells. So you'll see a range more typically than you'll see an exact number when it comes to the total amount that are made. All right, here's our diagram that is outlining the entire picture that we just studied. So we had these processes. We had glycolysis, we had pyruvate oxidation, and the citric acid cycle. These together are called the Krebs cycle. Oops, I'm sorry. Krebs cycle actually refers just to this one. My bad. So we have the Krebs cycle going on, and then we have this last stage, oxidative phosphorylation, which is the electron transport chain and chemiosmosis. So this diagram does a really good job of summarizing where the energy is flowing in this entire process. So when we say the energy, you're looking for these, NADH, FADH2, and ATP. The end goal here was to create ATP, so we've got a lot of it coming out of there. You'll notice that most of it is coming from this stage right here, oxidative phosphorylation. Krebs cycle contributes a couple of ATP, and glycolysis contributes a couple of ATP. The last section that we looked at was looking specifically at the stages of aerobic cellular respiration in detail. But remember, aerobic is only one of 3 types of cell respiration. So we can visualize the 3 types in this flowchart. We start off with a molecular energy source, meaning food. And then if we have oxygen present and the organism uses oxygen, then we'll be going through, that's supposed to be an O2, we'll go through aerobic. So that was the first type that we just talked about. And the aerobic respiration, you can follow these and read them, but I'm going to skip over them for right now. So if we have no oxygen present, then we're going to utilize one of these other two pathways. So we could use anaerobic cell respiration and fermentation. I said before, these two will get used interchangeably. Their only real relation is that they don't use oxygen. So they're two very different pathways. Notice at the end of them, or right here in the middle, we're talking about the hydrogen electron acceptors, and then at the end we're talking about the products. So this will be a good chart to return to. Like I said, for now we're going to zoom in on these two because we looked at the last two in the last section. All right, so these two types of cell respiration without O2. First one is anaerobic respiration. This goes through the same steps as aerobic. However, it has a different final electron acceptor, not oxygen, and it creates a medium amount of ATP. The estimates I'm seeing are pretty comparable to what aerobic cellular respiration makes, but typically it's stated that it will make a little bit less, or it's thought that. Fermentation, on the other hand, uses substrate level phosphorylation only. There's no electron transport chain, which means we have a really small amount of glucose, because the electron transport chain, this is the part that gets us the huge ATP payoff. Oh, and to make life more fun, fermentation can be divided into multiple types. So that process of fermentation is really glycolysis plus other reactions. There are two common types that you're expected to know. One is alcohol fermentation, so making alcohol. The other is lactic acid fermentation, which is making lactic acid. And this diagram does a good job of showing what's happening. So during fermentation, we start with our glucose molecule here. You'll notice that it gets split, which produces ATPs and two pyruvates. In this diagram, this would be like, oh, I don't know, a human muscle cell or a yeast cell. So in this diagram, if we don't have any oxygen, these pyruvates, if we are talking about a muscle cell here, these pyruvates will actually get oxidized to make lactate or lactic acid. And then in a yeast cell, these pyruvates would get oxidized to make ethanol and CO2. And so notice the ATP is generated back here just from splitting the glucose, and it actually takes a little bit of energy just to get back what you put in. So those are the two types of fermentation. Reason why we're going over those is because they happen to be very economically and biomechanically important for us. So alcohol fermentation takes pyruvate, turns into ethanol, that's drinking alcohol, and CO2. Alcoholic fermentation by yeast, this is used in brewing, it's used in winemaking, it's also used in baking. Only in baking, we stop the process before we run out of too much oxygen, so we don't get alcohol. All right, lactic acid fermentation, on the other hand, we have pyruvates, we turn those into lactates. Does not release any CO2. This is used by some fungi and bacteria to make cheese and yogurt. It's also used by our muscle cells to generate ATP when oxygen is scarce. So let's say that you've been sprinting for a few minutes and you're starting to breathe really hard. Well, that probably means that your cells are running low on oxygen. That's why you're breathing harder, to try and get more oxygen. When your cells run out of oxygen, that's when they start to do fermentation just to get some ATP, because your muscle cells can't stop working. They need ATP somehow. If they don't have oxygen, they've got to make lactic acid. Kind of a side note here, but in ecology, you're going to see that bacteria are labeled or named by the type of cell respiration that they use. And there's two types that we'll bring up right now. One is obligate anaerobes. So aerobe would mean that it uses oxygen. The an cancels that out. So these ones cannot survive in the presence of oxygen. They are anaerobic, no air. They have to use fermentation or anaerobic respiration. One example that you guys might be familiar with would be this bacteria, which causes tetanus. There's also facultative anaerobes. These ones could use fermentation or cell respiration. They can switch back and forth. So oxygen's not going to kill them. One example there would be yeast. There's also Staphylococci bacteria. That'd be the bacteria that causes staph infections. And then also E. coli bacteria. Okay, glycolysis, as it turns out, is pretty significant evolutionarily, because if we look at a large variety of organisms, they have it. So ancient prokaryotes, meaning cells that didn't have a nucleus, they're thought to have used glycolysis before there was oxygen in the atmosphere. Very little oxygen was available in the atmosphere, and it actually poisoned organisms when it was in the atmosphere. So early prokaryotes probably used glycolysis to generate ATP. And we're talking about 2.7 billion years ago, so we're talking quite a ways back that process has been around. So it's an ancient process. It's getting brought up right now because when we go to talk about evolution, we will talk about conserved processes, meaning things that date back to potentially the first living thing. And so glycolysis is one of those things that it's shared by lots of bacteria, it's shared by a lot of eukaryotes, so it's thought that this is a pretty important evolutionary step. All right, comparing and contrasting all three types of cell respiration. So we've been talking about these types, and now it's time to kind of zoom back out and review what we already know. So all three of these processes are catabolic and exergonic. They use the process of glycolysis to make some ATP. They also use NAD+ as an oxidizing agent, or as a battery, that accepts electrons during glycolysis. And all three make ATP. That was the whole point of them. Each process has a different final electron acceptor. In aerobic respiration, oxygen is the final electron acceptor. In anaerobic respiration, another molecule will be the acceptor, so not oxygen. That's why it gets its name anaerobic, not using oxygen. Fermentation, we'll use pyruvate or acetaldehyde to finally collect the electrons. And those make different amounts of energy. So aerobic making the most, anaerobic making a middle amount, and then fermentation making the least. All right, you have reached the bonus round. So if you are feeling confident with what we've gone over up till now, then the last thing we're going to add in is how we can use different fuels, so not just glucose, to do cell respiration. And in your book, it's referred to as concept 9.6. Okay, so if we want to use other fuels for aerobic cell respiration, we need to recognize that there are a lot of different ways to get these molecules into cell respiration. For instance, glycolysis will accept a wide range of carbs, not just glucose, but it could also work on other carbohydrates that we feed it. Proteins, on the other hand, they need to be broken down into amino acids, and then those amino groups can feed glycolysis, so they can be used to get split up to make ATP, or they could go into the citric acid cycle. Fats, if we feed those to the cell here, those get digested into glycerol and into fatty acids. So that should look familiar when we talked about macromolecules. Those fatty acids get broken down by this other process called beta oxidation, and they end up yielding acetyl-CoA. 1 oxidized gram of fat produces more than twice the amount of ATP as an oxidized gram of carbohydrates. And you guys probably remember when we talked about calories that we said that the typical carbohydrate is going to yield about 4 calories per gram. Same with the typical protein, and the typical lipid will yield about 9 calories per gram. That all comes back to this process here. Because this process happens to be much better at producing energy. This diagram does a good job of showing you kind of how different things feed into cell respiration. It's the last image that we've got to look at. And what it's showing you on here is the pathway that different macromolecules take to get into cell respiration. So right here in the center of the diagram, these are all cell respiration processes. And then here we have the different macromolecule types getting fed into these. You would not be expected to memorize this. I'm not going to expect you to memorize this. What I would expect is that you can read a diagram like this, and from that understand that proteins are going to get turned into amino acids, and then from there converted into pyruvate or fed into this cycle, however you want to say that. So you should be able to follow a diagram like this and make sense of it. Maybe if I ask you some questions like, what would be a waste product of protein metabolism or catabolism? Here we see that proteins get broken down. We see that NH3 is leaving, so that might be a waste product. I'm going to ask you, at what stage does it appear that fats will enter into cell respiration? You'd be able to read through here and see, okay, fats get broken into glycerol and fatty acids. Glycerol enters here, fatty acids enter here. So it's more about being able to read and understand, but you do need some of that foundational stuff in order for this part to make sense. All right, guys, that is the end of our very quick, almost an hour-long crash course on cell respiration. I hope that helps clarify things. Your journey is by no means done. Now the hard work begins of making sense of all of this stuff. Good luck.